I'm not sure if I want these online, but you'll presumably write to me about these. Yeah. There's some stuff, because it's not published, I would like to have it published before we...
Oh, I see.
Yeah, yeah.
Well, if you don't...
Yeah, I'll let you know. Both today and yesterday's will be online very soon, and I would just like to get that there.
Okay. Yeah. If you want to, don't forget if you want me to, it's fine.
Oh, it's okay. We can, it's good. Yeah.
Okay.
Yeah. All right, let's... Okay, wait, so we need to turn this on. And then it's...
Try to move.
Yes, it figured it. Well, good morning. Thank you for coming along. I'm sure you're tired too, much tireder than I am. Tired of biology, goddamn tired of Rainey. So, common theme. The idea was a set of lectures on a theme. I'll change that this afternoon. Again, scaffolding, individuality. I really don't need to say much by way of introduction, but what sets this one slightly apart is the fact that the focus is not on those fraternal transitions, such as the one that leads to multicellular life from individual cells. That's a fraternal transition. Here we are thinking about transitions that involve separately replicating entities. And again, I've emphasized a number of times, well, we'll see a picture in a moment, a nice example of an egalitarian transition is the evolution of the eukaryotic cell, where two separate cells came together to form an alliance, and in forming that alliance, aligned their reproductive fates. So we had that same theme. This is sounding very loud. Can I turn it down? It's okay? Okay. What happened there when two cells became one is their replicative fates became linked, completely aligned. And when that happened, that solved the problem of heredity, and that's what we're going to look at today. Rather like yesterday, too, I will describe my intuition of what might happen if we were to do this hypothetical experiment, an experiment that we are doing. And that's led to some theory to explore the validity of those intuitions. So it's largely based on theory. We don't need to go through this again. It sets the scene. I've mentioned it twice. The issue that we're dealing with, again, is how do these Darwinian properties arise if we set the bar high and take nothing for granted? And actually, if we do set our sights on thinking about evolution of the eukaryotic cell, it is actually very difficult to imagine what happened that led to the stabilization of this alliance. So scaffolding, again, you know about this. So let me say a little bit more about this. I showed you this slide on Tuesday, pointing out that there are two kinds of transitions. This is David Queller's insight, and it's useful, I argued on Tuesday, because these different transitions define different problems. The fraternal transition, the evolution of multicellular life, begins with one cell, and we would often think about the seminal mutation being one that causes the daughter cells to remain attached in some way, just as you saw with the sticky mats, those wrinkly spreader mats that I mentioned on Tuesday. And that mat population grows, and the problem that needs to be solved in terms of those key Darwinian traits is reproduction. To a large extent, heredity is solved by the fact that we have one cell that replicates itself, and that one cell, of course, already has genetic material that obeys the laws of Mendelian genetics. So heredity is basically solved in fraternal transitions. What needs to be solved is a means by which the group reproduces. That group, as I pointed out again earlier on, if it simply gets bigger and bigger and bigger, there is no evolutionary process happening at the level of the collective. So that collective needs to find some way of reproducing. Typically that's through a single cell bottleneck, reestablishment of the multicellular state from that single cell bottleneck. In many forms of multicellular life, for good reason, early on, the germline is sequestered. Of course, this involves a developmental process, and the origin of that sequestration event is tremendously interesting in itself. And so once we have that soma germline, we have a germline that is effectively aided by the soma. And we saw that in the experimental studies on Tuesday and yesterday in the theoretical studies that showed, given a certain ecological structure, conditions are set for the evolution of this reproductive division of labor. Those cells that proceed to the next generation, the evolutionarily relevant cells, and all the rest, which are like those B cells, if you remember yesterday, that are simply there to aid the germline. And that, sadly, is the story of us. Or, for good reason. These egalitarian transitions, the problem that needs to be solved here is not reproduction, because the two entities have that capacity already. What they don't have is an alignment of reproductive fates. So what needs to be solved there is heritability. So let's imagine that these red and blue cells, by chance, happen to come into one another's proximity. And let's imagine the possibility that there is some metabolic cross-feeding. So a waste product from the red cell becomes food for the blue, and vice versa. So there is some mutual dependency. And that's easy to envisage. That sort of thing happens commonly simply by chance. So that all looks good, but that alliance will be unstable. Selection will always favor mutant types that take advantage of the other. And that certainly threatens the alignment. So what needs to happen for these two entities to be able to recreate? I guess I haven't said this. When I'm thinking about heredity, I'm thinking about a property of the community here. I'm thinking about that property being stable. So without this alignment, that property is not going to be stable. One recursion of this in one isolated instance may have roughly a 1-to-1 ratio, and the collective has a good phenotype. But somewhere else, because a mutant, let's say in the red type, means that the red type is taking unfairly of the resources of the blue type. So there are very few blue, there are many red, and the collective phenotype is lost. So we're thinking about how that heritable, how the phenotype of the collective becomes heritable. And so this is the problem that needs to be solved here. The eukaryotic cell, as I've pointed out a number of times, involved one cell engulfing another, or alternatively, one cell invading another.
In the example you mentioned, why do you say stable? So if the red guys are more than the blue ones, they produce more, say, goods for the blue ones. So the blue will be advantaged. They will grow.
Well, it could be that way around, or it could be that the red is taking so intensely of the products of the blue that blue is deprived resources to grow itself. So blue is excluded. It could go either way, but it will definitely be unstable. Yeah. So how these things become stabilized is what we're going to talk about today. So there is the eukaryotic cell. Stability of the phenotype, the heritability of the phenotype, was assured, more or less, upon this engulfment event or invasion event. If we think about genes becoming members of a chromosome, again, separate DNA molecules, they could interact, but again, it'll be unstable. But if they become linked on the same chromosome, their fates then become linked. Their reproductive fates become perfectly aligned. Oh, and indeed, here is an example of an egalitarian transition. This is one hypothesis from Eugene Koonin and Bill Martin, involving here successive stages of engulfment, with an archaeal-like bacterium engulfing a protobacterium that then becomes the equivalent of the mitochondria, well, becomes the mitochondria, with many other events that need to happen en route. But that, in essence, is the transition involving two cells becoming a single entity. Two genes becoming a single entity, as I just mentioned on the chromosome, is another egalitarian transition. And I made some comments yesterday about other kinds of transitions that we could envisage, such as those between humans and artificial intelligences. And I made the point that it doesn't need to be, it's not in the realms of science fiction. These sorts of transitions could indeed come about with the same sorts of consequences as we see for transitions such as these. Two entities that were once separate aligned their reproductive fates with us as humans being effectively slave to this higher level of selection. And that raises some very interesting, it's kind of, there's a science fiction novel in there that's almost too real to joke about. As I pointed out again previously, that this idea of scaffolding is really about recognizing the potency of the process of evolution by natural selection and realizing that this can be conferred from the outside. So again, we see an example here applied to egalitarian transitions. Here we've got a population of cells. Colors indicate that they're different, so it's a community of cells. What we can do very easily, we can do it with a test tube, we could do it with a droplet. We can draw a boundary there, and we can get rid of the cells outside of them. And simply by putting a boundary around those cells, we've discretized variation into a set of particles that are within a black sphere and within a red circle. So we have variation, and it's discrete, simply by putting boundaries around these entities. These Darwinian properties down the bottom here, we could allow a reproduction event, a birth-death process perhaps, to take place now at the level of these test tubes, droplets, as I'm going to go on and show you. We decide that, or it could happen in the environment, but here I get to play God. We decide that this community is really not performing, the function that we're interested in is minimal. So we mark it for extinction. On the other hand, this collective turns out to be doing, some metabolism that's really interesting. So we say, we'll keep you. And not only will we keep you, but we will allow you to reproduce. So that simply involves splitting this population in half and distributing the contents into two new types. So we had a variation. There is a reproduction event. And now heredity, and I've already pointed out that the alliance between cells and their contribution to the collective phenotype is what we are interested in. So here there is perhaps we might think of as a kind of a marginal sort of heredity. I'm considering, let me just go back here, we put those boundaries around. I'm now imagining that these cells within these test tubes, for example, are growing. So this population of cells comprised of these types is now very large. When I divide it, what I'm thinking of doing is taking a sample of cells and establishing them in two fresh populations. So I take a sample, and so the stochastic process of sampling will have a primary effect on the composition of cells at this next stage. And of course, there the bottleneck size is important. If I took a very large sample of cells, then we are likely to reestablish the phenotype. But if I took a small sample of cells, it's very unlikely that the offspring phenotype will match the parental type. So the intuition here, and I'll try and get this across, is that interactions, perhaps not surprisingly, are the basis of heredity. So when we were thinking about evolution of the eukaryotic cells, two cells coming together, that becomes initially an interaction due to metabolism, perhaps. But once one is engulfed or has invaded the other, at that stage, we have an extremely intimate interaction. And in fact, it's basically an irreversible interaction. So why, well, I guess the question then comes, how do these interactions emerge? How could scaffolding lead to the evolution of these interactions? Because that's what I think it will do. If we treat these populations as Darwinian entities by our imposition of these properties on these test tubes or droplets, then through time, if we continue this process, we will build interactions between cells that will stabilize this community. Now, why is that? Well, let's go back to this process of growth within a patch. Growth of all of those cells, we get a population, let's say, of 10 to the 9 cells comprised of different types. And we measure the phenotype of these test tubes or these droplets. And we find this is a great droplet, this is a poor droplet. So we mark the poor one for extinction. Now, there are no interactions at this stage within the droplet. We've identified a great droplet on the basis of some phenotype we're measuring. So now we dilute. And again, let's imagine we dilute to a very small number of cells. And as I pointed out before, the offspring droplets are very unlikely to resemble the parental type. So in the next generation, although the parent was great, the offspring are rubbish, and when those offspring have grown up, they're rubbish and they're eliminated. So we start again. Imagine now that there is an interaction that becomes established that stabilizes the ratio of, let's say, 2 types. Now we dilute to a low number. Let's say red types stick to green types now. So we don't dilute to a low number, and the good droplet had a 1-to-1 ratio. Red now sticks to green. We dilute down to 10 cells, but because red sticks to green, we have a 1-to-1 ratio at the offspring stage. It grows back up to an adult, large population size, but still a 1-to-1 ratio. So it goes on, it's chosen next time. So the process of droplet level selection should favor the evolution of those communities that evolve interactions. So that is effectively what led to the following work. So the way that we scaffold these populations, it could be done in test tubes, or easily it can be done in test tubes, but one is thinking about perhaps thousands of replicate populations and transferring every day and assaying thousands of replicate populations becomes a daunting task. You perhaps know about, microfluidic technologies where enzymes, chemical reactions, or individual cells are captured in an emulsion, in a droplet of liquid within an oil background. The millifluidic technologies, actually you saw in the talk at the end on antibiotic resistance yesterday, a kind of millifluidic device based on a mother machine. What we're thinking about here are emulsions where we've got basically salad dressing, droplets of liquid in a background of oil. So oil and then, water. It could be media. These droplets are typically picoliter in size, so one only has a small number of cells in them. The advantage of this is that you can generate millions of cells millions of droplets in an extremely short period of time. The rate is something like 10,000 a second are generated with very simple technologies. Some years ago, I guess about 5 years ago, where I began my career shift back to Europe and spent time in Paris at ESPCI, where I still retain a lab, Jérôme Babette had established a technology rather than microfluidic. We call it millifluidic. The droplets are in the nanoliter, actually now up to we're up to microliter. We can now get up to 10 microliter sized droplets. So these are large enough to be able to maintain large populations of cells. In a microliter, you have around 10 to the 6 cells. So around 10 to the 7 cells in a 10 microliter droplet. One can start doing population biology with population sizes of that sort of size. So this kind of technology has been put to work. There is now a spin-out company, and this shows inside of this device. What you will see is that the emulsion is very carefully controlled. In fact, you can see here, I'll show you a movie in a second, there are 2 coils there, and wrapped around the coil is around 5 meters on this side and 5 meters on this side of Teflon tubing. And the droplets are maintained in an ordered array inside of this tubing. So media for growth of the bacterium. This is a plug of air, and around it is a fluorinated oil. So it's an emulsion, a controlled emulsion. By controlling the pressure, we can move these droplets. Well, first of all, we can establish a train of droplets, and you'll see this in a moment. And then this train of droplets can be maintained in this coil, moving backward and forward at an appropriate temperature, during which time growth takes place. Actually, it looks very different these days, this machine, but this is sipping to load the train. So there are bacteria or microbes of some sort in those wells. Let me just stop that for a second. So here's a 96 well micro titer plate. One could use a 384 or larger. Here is a little sipper. It is sucking a sample of cells and media to make one of these droplets. It spends a certain amount of time sucking. There is oil running down the outside, which is also being sucked up. So the process of loading the train is also allowing the emulsion to form. So as a result of this sipping process that you see, a train of droplets and air and droplets is established, very simply by controlling the length of time that the tip is in the microtiter well. So if it's in for a short period of time, brings in oil, a droplet, and as it's out, it sucks in air and oil, and then it goes back in and samples once again. Whoops. And here you can see the little droplets here. They've got color in them, so you can see them. There's the plug of air and the colored droplet. So large enough for you to see. These are about 500 nanoliter droplets. These droplets are now being incubated. They've been filled. They pass in front of a laser and a microscope, so one can pick up any sort of fluorescence signal. There are multiple lasers here to detect any fluorescent signal you want. Train has got to the end and it's coming back, so as each droplet passes through the light beam, information is picked, so we can measure the phenotype of each droplet, and one can do all sorts of things with these droplets. Here is feeding some new resources into the droplet. There it's picked up into the droplet. That's the plug of air. Here is at the opposite end. We've got, there's been growth in the machine, in the droplets, and we've assayed the phenotype of those droplets. It could be some histochemical reaction that's coupled to a fluorescent signal. It could be some chemical activity, could be frequencies of different types, but we've made a decision now on the basis of those phenotypes that have been recorded during the process of incubation as to whether drops will live or die. And this just shows the process by which the initial loading, the sipping, is reversed. So the droplets are now being spat out in an ordered array, and they're either spat to waste or they're put into a fresh microtiter plate where there are no bacteria, but there's fresh media. So a droplet of now 10 to the 6 cells is put back into a droplet, into a microtiter plate. So this now has growth in it. We say, you know, you're a winner, you're a loser, and that droplet is, well, we have you know, a bin for waste, and we have wells in which we collect the good guys. So this is spat out into here. This contains media. 10 to the 6 cells then are diluted in whatever volume you like. So that, let's say that's 100 microliters of liquid. And then that one goes to waste. The next one might be a good one, and it's dropped into here. And again, diluted. From that, we can reverse the process. So we could say, this, again, it's been a great well, and because there's been a death event, this one was eliminated, we're going to allow this droplet to reproduce. So that simply means when we re-establish the train, we sip once and twice. And that means that droplet's now left 2 offspring droplet. So there's been a dilution and a resampling. So this is Guillaume Doucier, who is a rare breed. Not only is he exceptional, but he started life as a biologist, and he's turned into a mathematician. The thought experiment is one that takes a community of red and green bacteria. So here we're thinking, label one bacterium, you know, it could be E. coli, or our favorite Pseudomonas with GFP, green fluorescent protein. Label another, isogenic, with a red fluorescent protein. Let's imagine we establish a 1-to-1 ratio inside of a droplet. So, well, just to remind you, first of all, that what we're thinking about is how we align the reproductive fates of these 2 types. So we establish a population with a 1-to-1 ratio of red to green. Now we need to select or think about a collective level property, and that collective level property is a combination of the color red and green, the color brown. Now we can select on the color brown simply by measuring the relative ratios of green to reds in each droplet. So we have a collective phenotype here. We're not going to be selecting on green cells or red cells. To be successful, you need to be a 1-to-1 ratio, which gives us the color brown. To make the connection to something I said earlier, the life cycle experiment, I made comment about the fact that selection there, once we had that 2-phase life cycle, was seeing a developmental program that it hadn't seen before. Selection needs to work on something other than the properties of the individuals. Here, we are rewarding those droplets that have a collective phenotype that is a combination of 2 types. But that could be some chemistry. It could be anything you like. For the thought experiment here, it is a color which is the combination of the 2. Now, I'm going to start out talking red and green, but during the process of writing this paper, that will be on the archive very soon, it was pointed out, of course, that among males, green and red color blindness is not uncommon. So we've switched to red and blue. So when I switch to red and blue, it doesn't matter. We're just still thinking about a combination of colors. Yeah. Yeah.
This is abstract. Is there some barrier for an individual to co-opt the property of some other individual and on its own produce what you are calling a collective property? Meaning, you have 2 players together, they make some product that then is the collective property. If one of those, say, a gene from another bacteria transfers to me, all of a sudden I have all the properties.
That's right. That's an entirely possible outcome. Perhaps one reason for doing this experiment. I must say, as an experimentalist, after I go through what Guillaume's done, I almost think there's no point doing an experiment except for those surprising outcomes. So yes, indeed, that would be a great way of solving this inheritance problem. If GFP was transferred by a transformation event from the green cell into the red cell, so we now have a single cell that expresses the color brown, well, there you've affected the stabilization of reproductive fates. It's done.
Yeah.
Yeah.
So you didn't do anything to stop that.
No.
In advance, you did something.
No. That would definitely be a possible outcome. In what follows, you will see from the theory, we have just allowed density-dependent interactions to emerge as the only way of building interactions. And you will see the outcome is quite surprising. But indeed, there are many ways, additionally, that you could build interactions by sticking, by acquiring a gene, by engulfment of a whole organism. So again, here is our... well, this will start as a video in a second, but here is the thought experiment. So we're imagining founding multiple droplets with a 1-to-1 ratio of red and green, giving them a period of growth, say 24 hours, 48 hours, it could be what we wish. During that period of growth, of course, there is evolution within a droplet. And what I'm showing you here is that after a period of growth, these same droplets have a different phenotype as a consequence of mutations arising within each type. And in the models I show, these mutations are affecting rate of growth, and actually they're also affecting the nature of density-dependent interactions. But if we just thought about mutations affecting rate of growth, we now have a distribution of colors that we can assess with our machine. And we've said, look, it's a 1-to-1 ratio that's the winning type. If you're too far away from a 1-to-1 ratio, then you will be marked for extinction. And the place you occupied will be available for types that avoided extinction because they were within the boundary. So what you have here, of course, again, is a nested Darwinian population with 2 time scales. So exact parallels with what we've seen before. So the time scale of the doubling time of cells, and then the time scale of the doubling time of droplets. And again, you will see that the time, whether it's long or short, is tremendously important. The types chosen for reproduction are chosen at random from those that exist. We don't choose the ones that are closest to perfection. And in fact, choosing at random is much better than choosing the best ones, which is always interesting. And that takes place within one of these droplet chains. So again, there's the big plug of air. It keeps the droplets separate. The smaller droplet is the droplet-containing media. And again, it's established via the mechanism I mentioned. And once that train is made, then it goes back and forward. That's the growth phase. Okay, so that's the thought experiment. Guillaume did some math. He starts out with a stochastic model to describe behavior within inside of droplets. For those that know about these earlier studies and the stochastic corrector model in particular, there are similarities to it. But basically, we have a nested Darwinian process where there is a birth and death process happening both at the level of cells and at the level of droplets. So this is an overview of the stochastic model. What matters? We have a color. Okay, now we've switched to red or blue, not red or green. But cells are either of 2 types, and that's fixed. Red cells cannot change to blue, and blue cannot change to red. There is a growth parameter, and then there are 2 parameters that define competition within a color and between a color. And that allows the following events to take place. So particles divide, grow, and produce offspring at a particular rate. So that birth rate can be affected by mutation. And well, let me come back to the evolution part. Additionally, we can have within color competition, which can bring about death. We can also have between color competition that brings about death. later, I will show you that these are approximated using, again, a Loc-de-Volterra approach. So evolution affects the dynamics within these droplets. The growth rate can vary stochastically. Mutations affect the growth rate, and the interactions between red and blue types is also affected by mutation. I think I've explained this in the introduction, but just to make clear what's going on. Here is the establishment of an offspring droplet, which has been sampled from a parent. There are a small number of droplets there. There is a period of growth. At the end of the growth phase, we assess the phenotype of the droplets, and we ask, is it within a certain ratio, certain bound of a 1-to-1 ratio of colors? If it is, the droplet is in the game for the next generation. If it's too far away, too blue or too red, then the droplet goes extinct, goes to waste. The space in the train of fixed, which is fixed in terms of number of droplets, is therefore, there are two opportunities, two fresh droplets that can be occupied. In this instance, this droplet here, which met the criteria for persistence, was split into 3. Again, that splitting event involves a dilution phase, where we have a lot of stochasticity in the absence of interaction, and then a growth phase. Okay? If we run that experiment without imposing selection at the level of droplets, then as you would expect, very, very quickly, either blue types fix or red types fix, because you will get, the interaction is unstable. Mutations will arise. If it arises in the red and it increases growth rate, red will fix. Similarly in the blue. So within a very small number of generations, we've lost our community. So what are you seeing here? The y-axis here indicates at the top droplets that are all blue, all red. This is a result of the simulation. Populations were founded with a 1-to-1 ratio. The circles, you can just make out red and blue is the proportion in each droplet, so all blue, all red, or somewhere in between. And the branches indicate offspring droplets, or the connections. So there's a genealogical connection between them. And what you can see is, although this is the ideal 1-to-1 frequency, we're not selecting for that. We're killing off 20% of the droplets each time, but we're choosing the droplets that reproduce at random. So there is no selection at the level of droplet phenotype. And very quickly we fix either blue or red. So it makes sense.
So when you repeat the experiment many times, the number of fixation of blues and reds are the same?
Well, no, because we start with red particles that have a faster growth rate than blues. But I would expect if and we do that in order to kick the system into action. Otherwise, I would expect, yes, that blue, unless we have some asymmetry in the properties, in the genotype of blues or reds, it should be either blue and red will fix with equal probability.
Because you began with 50-50%.
Yes.
So you expect something like an balanced phenotype. So you expect the fixation to be the same.
Yes, I would, for blue and, yeah, exactly. And yeah. And I'll show you some more detailed analysis of the neutral model in a minute.
Two slides before,
Yeah.
I think I saw a dark blue. Yeah, here.
That's to indicate, so the color is not changing, it's just that the growth rate has changed. That's what the color is intended to indicate. So actually, if we come back to here, you've got different shades of red. That's because that's intended to indicate The growth rate, yes. So perhaps I didn't come back to this, but so each particle has a genotype, okay, which is determined by growth rate and interaction rate. And that determines the phenotype of the cell, and of course, collectively, it determines the phenotype of the collective. And so later on, we'll delve into the relationship between the genotype and the phenotype to see what goes on. So here we saw, without droplet level selection, we very rapidly see fixation of either blue or red. If we now impose droplet level selection by the means that I explained, and what we're doing each time is eliminating the 20% of the population. I think we have 1,000 droplets here. 20% are eliminated, and that allows the possibility for a subset of extant droplets to reproduce. And when we do this, here is an overall timeline, and then we see it broken down into the first 50, and then generations 250 to 300, and I'll show you a nice movie from Guillaume in a moment. What you can see is that, well, if we come up here, this is over, I think, 10,000 generations. No, this is less than that, but we usually run it for 1,000 generations. What you can see, starting at the beginning, is this very high variance. And as time goes by, the variance decreases. So droplet level selection seems to be having an effect. And of course, the most obvious effect might be relatively trivial, that by forcing things in this way, we may simply be forcing alignment of growth rates, for example. It's just extremely hard selection. Now, if that were the case, if we were to stop selecting at the level of droplets, we would immediately see blue and red fix within quick periods of time. If something else has happened, and there is the potential for that because of the density-dependent interactions, we would be much more interested. So let's see what we find. It looks good here, though. Here is actually a snapshot. We're back to green and red here. As time goes by, and you know, I mean, Guillaume likes doing things with not only numbers, but representation, and he does a fine job, actually. So here you see, as we get toward the end of this experiment, that most droplets are giving rise to offspring droplets that resemble the parent, or grow up, I should say, to give the parental phenotype. So in thinking about heredity, actually, it's quite important. If we are to ask about the heritability of traits, so I'm talking about parent-offspring relationship, but if we want to know whether our children resemble the parents, we wouldn't measure the baby compared to the adult and say, there's no resemblance because the baby is really short and the parent is really large. We would wait till the baby has grown up. So when I'm talking about parent-offspring relationships, I am recognizing a developmental process, perhaps, one in which the parent has one phenotype, the offspring has a phenotype that perhaps doesn't resemble the parent, i.e. it's really small, but by the time it's grown up again, it does match the phenotype of the parent. And that's very relevant here. That's right. These are the parental phenotypes at the time of assessment. So what you see up here, 2 green or 2 red, been marked for extinction. There's no line that connects it to the next generation. Whereas once you're in this realm here, there are connections to the future generations of droplets. Droplet level selection does have consequences for the evolution of particle traits. Here we have droplet level selection going 10,000 generations, and again, what you're seeing is the density of droplets, large variance, it's just what you saw before, and a much reduced. So most droplets at the end here have this optimal 1-to-1 ratio. What you see, though, is additionally that growth rates change. So here you can see at the beginning, red has a higher growth rate than blue, but during the course of the experiment, growth rates go up. Well, they change, they go up largely. But so too, importantly, do interaction rates. Now, this may seem a little odd. I find it difficult, and it's something we need to sort of explain a little. But because we start with red types that have a faster growth rate, the interaction rate is primarily affected in the red type because it's growing faster. It's changing a little, but not so much from the slower growing one. But that simply reflects the initial state of the system. So this is the rate at which cells engage in density-dependent effects. So the simplest way to think about that and the way we factor it in is some sort of allelopathic effect. So that as a cell produces a toxic compound, for example, when it enters into stationary phase, and that inhibits the growth of another. And it's the rate at which that phenotype is affecting other cells within the population. So the greater the rate, the greater the effect. This is all done mathematically. In a moment, we'll go to some deterministic approximation to try and see what's going on. Exactly.
But is interaction dependent on color?
No, this is between. This is just between colors. We don't allow within color to evolve. We allow the possibility of interactions between the different colors to evolve. Here's a snapshot of what's happening within the droplets. This is the result. This is, in fact, the gray lines are the trajectories of every single droplet, and this is the mean. This is at generation 1. This is the proportion of red cells. And you can see it's all over the place. By generation 9000, though, where we've been selecting for a 1-to-1 ratio, you can see that there is a very different shape to the dynamics within these droplets. And we'll look more into that in a moment. We will go on to some ODE approximations. The solid lines are the results of the stochastic process, again, at generation 1. This is the number of individuals within a droplet, so they are just growing, reaching into exponential phase. By generation 9000, that dynamic is quite different. And this is, well, I will emphasize this a little more because this is interesting. So the growth rate chosen means early on they aren't reaching stationary phase. At the end of the growth period time, within a droplet, at the time that these droplets are going to be assessed and split, the cells are still in exponential phase. And you perhaps recall, I'll show you again, that during the course of this experiment, growth rate increased. And that growth rate increase means that what selection has favored are droplets where the cells enter into stationary phase earlier on. And it's the stationary phase where the density-dependent interactions become relevant. If you have too low a population number here, and note the different number of individuals, the scale is different, there is little opportunity for density-dependent interactions, and that's the basis of the interaction that can evolve. If after 5,000 generations of droplet-level selection, we now stop droplet-level selection and ask what happens, so we continue the process, but we no longer reward droplets that are closer to a 1-to-1 ratio, we just choose at random. Earlier on, I showed you that at the start, if we don't impose droplet-level selection, then blue or red fix within about 10 generations. After 5,000 generations, we stop droplet-level selection, and we see that even 500 generations later, this phenotype is heritable, or persists. And of course, what one wonders now is the mechanistic basis of this, what's gone on. And what that does require is a way of mapping particle traits, particularly growth rate and interaction rate, to droplet-level phenotypes. And again, Guillaume has done this, drawing upon, once again, good old Locke de Volterra competitive models. What this shows is what's going on inside of the droplets at generation 1 and at generation 9000. what you see here is a series of iterations. So, Droplet is founded with a small number of cells. It grows up. That's the solid line. And then it's diluted back down. And then that same droplet grows up again, and it's diluted down, grows up again, diluted down. And we end up with fixation of the red type, because red types grow more rapidly. There is no internal stable fixed point. There is a fixed point only at the boundary where we have fixation of the red type. After 9,000 generations, though, it looks very different. Again, we allow growth, and it comes to this stable fixed point, internal fixed point. We dilute, goes up around, and always descends to this internal fixed point. Another way of looking at this, so what we were faced with here is trying to get across what it means to think about and measure the evolution of heredity. We can take this information and plot it slightly differently, and we can get this out of the ODEs as well, but for me this is really intuitive. So to begin with, what we have here, let's say, is a droplet that has this particular ratio of green to red. This is the red cell. We allow it to grow up. Because red grows faster, the parent, once it's grown up, has many more red cells. Okay, so we take 0.65 and we start that process again. Okay? Red fixes. The shape of this line here we refer to as the G recursion or the G function. And it's this that evolves through time. So to begin with, there is basically no heredity. We see that. We get fixation of the red type. And that's captured in the shape of this line. Okay. Once we get to this point, though, after 9,000 generations, the shape of the G function is very different. And the difference between here and here is all about the evolution, or our attempt to capture a measure of the evolution of heritability. And what this means, once we're in this realm here, is that it actually doesn't matter within a large range what the proportion of blue and red types are at the dilution time. They come back to having the phenotype that is optimal. So we can measure, so this provides a measure of heritability of the system. I meant to put in a slide of canalization from Waddington. It is as though there is a developmental process that's emerged here as a consequence of the interactions. Okay, we don't need that.
So this bottom right one, that is
9,000 generations.
Is that empirical, or that's the model?
That's the result of the Lochte-Volterra approximations of the stochastic system. Yeah.
So, I mean, when you get to this phase, maybe some people recognize this, these are like all the way to single discrete maps. And so this, I don't know if you made an extreme where it's here, but as long as that slope, because you have the first, you have the current proportion, the next proportion, it doesn't even have to be flat. Anything
that's right.
Not too steep will get it back and stable. So it's quite robust.
It is, and we'll look exactly at the dependency now on the duration time of dispersal, and actually look at its evolution through the course of this experiment, because this really does evolve during the course of the experiment. And indeed, once you get to the stage, it's pretty extreme here. It's very robust. I showed you earlier on that 5,000 generations, it will go on. We can stop droplet level selection. It goes on for 500 generations. I'll show you in a moment. We go for 10,000 generations, and it goes on robustly. It's very difficult for mutants to invade once you've got to this sort of flatness of the G recursion. So as we saw yesterday, this is dependent on the time frame of the dilution event. here we have a relationship between the duration of the growth phase, so we haven't looked at varying that at the moment, and whether or not we see an internal stable fixed point emerging, and at what time that emerges. So if we have a very, very rapid dilution phase, so the second time scale is very rapid, there is no chance for the emergence of density-dependent interactions, and that makes sense because population density is low. Once we get to a critical point, progress then moves towards stability and the realization of this internal stable fixed point, which is the one-to-one ratio. P is the equilibrium of the ODEs, the Lochte-Volterra ODEs. What you see here is the impact of bottleneck size on emergence of this stable fixed point and its relationship to the duration of the growth phase. So what you can see, so up here in yellow, we've entered the stable fixed point. This is the analytical result, unstable down here, and you can see that bottleneck size does matter. The larger the bottleneck size, interestingly, the greater the range of stability. This is I find this very difficult to explain, and I think we need to think about this when it comes time to submit the paper, but what this really is the change that is happening during the experiment, in silico experiment, in the fixed point of G and in the growth phase. So these are things that do evolve. What you see here, so this is the critical fixed point. The populations don't achieve that for some 7,000, 6,700, 6 to 7,000 generations of growth. You need to look at this in context of this. This is actually the growth phase duration. So the growth phase duration is not changing during the course of we're not altering that during the course of our experiment, but it is being altered as a consequence of evolution within the droplets. And what you're seeing is these cells start out with a very fast growth rate, and that slows down. That allows for the emergence of density-dependent interactions to emerge, and that corresponds at the point where we get the stability. So now, if we remove selection after 10,000 generations, so at the beginning, here's our G function, basically a diagonal line. There is minimal heredity. At the end of the experiment, the shape of the G function has changed as a result of changes in the particles that themselves have responded to particle level selection. If we now eliminate selection at the level of collectives at 10,000 generations, you can see even 1,000 generations on, we've still got a distribution of droplets that have basically a one-to-one correspondence between particle types. It's immediately lost in the first generation. So something approximating heredity has emerged, and it's emerged through density-dependent interactions and their interaction with growth rate. This is kind of fun because we can pull out cells and see what happens. So this is what happens early on. That was the dilution and growth at generation 1 for a set of droplets chosen at random. And here at the end of the experiment, 10,000 generations, these density-dependent interactions within each droplet are returning particles to a one-to-one ratio. So wrapping up, what's really gone on here? One can think about it as a kind of a bootstrapping process. We could think about this phase space where reproduction here is possible. It's impossible outside of that state. So we've started with droplets with red and blue, red and green, red and blue, with a one-to-one ratio. Outside here, reproduction is rather disruptive. We can force it, though. We can force this to happen via our machine. The next stage is we see a response, which is evolution of properties within inside of the particle, which feed back to recreate the phenotypic space of the ancestor. And this reestablishes the parental phenotype. It's the interactions that have emerged here that do that. So heritability, the evolution of interactions, again, can emerge through this process of scaffolding. So those critical Darwinian properties, variation, reproduction, heredity, again, it's here what we're interested in, and what the challenge to explain in these egalitarian transitions is the emergence of heredity. And we see that in this fairly simple model, where the only kinds of interactions we've allowed are density-dependent ones, and there are many more kinds of interactions that in the real world could emerge, those interactions are sufficient to create a parent-offspring relationship that I'm comfortable calling the basis of heredity. There is indeed always much to think about. Again, one wonders about nature and whether these kinds of scaffolding events ever took place in nature. We can certainly do it in the lab. We can have a stab at recreating endosymbiotic events. But it's hard to imagine in nature the kinds of conditions that we see here operating. But perhaps, maybe we talk about that after having a break. And certainly there are applications for biotechnology, medicine, and agriculture. There's a lot of fundamental stuff to come from here, as well as these applications. So this is the end of the third lecture, at which point we draw to an end discussion of the evolution of individuality. We started off recognizing that life has this hierarchical structure with replicators nested within replicators, in the sense that this hierarchical structure evolved as a series of what has been referred to as major evolutionary transitions. They're perhaps not as mysterious as may have seemed on first talk, on my first presentation. I hope particularly after yesterday, you can see that the common theme between today and yesterday, timescales are critically important, the second timescale being absolutely critical. There being ecological conditions that allow a birth-death process at 2 timescales. And then the longer holds the shorter to account, and much can happen as a consequence of that, including the emergence of this hierarchical structure. And I've pointed out that one of the challenges, to me, the seminal challenge in this whole area, what really needs to be explained are the origins of these Darwinian properties. They can't be taken for free. And we've looked at how reproduction might emerge in the fraternal transitions and how heritability or heredity might emerge in these egalitarian transitions. And it is the case that Darwinian properties can be scaffolded. And we've seen another example here that these exogenously determined properties have become, to an extent, endogenized. Heredity, we can take away that scaffold. And even in the simplistic model, interactions are sufficient to maintain phenotype for many generations into the future. So those properties have become endogenized. And again, a final comment about life's hierarchical structure. It's really, I don't think that's surprising. All that is required are a set of ecological conditions where collective living is advantageous, be that as a community or as a single cell that gives rise to a collective of identical cells, as in the fraternal transitions. There are many instances where collective living is advantageous. What is necessary to go forward for those collectives to then become Darwinian entities is some means of recurrence of the collective phase. And that may be as simple as dispersal to a new environment where variation is maintained in a discrete state. And out of that can emerge quite simply a new hierarchical level. Now, of course, if it were all that simple, we might have expected much more complexity. My sense is that actually these ecological conditions are rare. And that said, actually, I should make a comment that I haven't said. The eukaryotic cell, when I was wondering whether scaffolding might have been relevant to its evolution, maybe. maybe there were very special conditions, because the eukaryotic cell has evolved, that evolutionary transition has occurred just a single time, whereas transitions to multicellular life have occurred on multiple occasions. Someone told me recently that the estimated number is more than 30 now. That seems to change. So becoming multicellular, which has happened in all domains of life, seems to be reasonably easily achieved. The eukaryotic cell, though, seems to be a really rare event, and what it ultimately required is unclear. And with that, let me just come to the acknowledgement slides again. So Guillaume Dossier, a PhD student in Paris, has done much of the work, and he is co-supervised by Amaury Lambert at the Collège de France, and Amaury is a real mathematician, and also involved and co-supervisory is Sylvia de Monte, a physicist at École Normale. Thank you very much. So we take coffee, I believe, yeah.
Okay, question session. Okay, I think I need a volunteer to hold the microphone in that aisle, so I stay here, someone else stays there, otherwise I have to run up and down. So, okay, thank you. The medallion one.
Okay.
Testing? Okay.
Go on.
After some thousand generations, we could see that the proportion was being, the 1-1 proportion was happening in most of the patches, you could say. But not through a symmetrical growth rate, but through a higher growth rate from one of them, and different interaction rates on the other hand. And they were like compensating each other, so that the final generation, the final population would be 1-1, right? But do you think that after enough generations, maybe tens of thousands, the growth rates would be symmetrical? And interaction rates also? Or do you think that was not...
I'm not sure. Yeah. That's something that bothers me. I say this to Guillaume, because it doesn't look great seeing... but he says there's nothing untoward going on. And it is a consequence of the initial conditions. And actually in the supplementary, he runs it with the, changes the conditions and shows it doesn't. I mean, that's the outcome we saw. And of course, it's a very simplistic model. There are very few assumptions governing the interaction. And that's, so at least through the simulations, there's many thousands of simulations that go on. They all generate overall the same sort of effect. Whether we would see anything different, I honestly don't think so. I mean, it's gone long enough. But, that would be another reason for doing the experiment, to see. As I pointed out, there are many more possibilities for interactions to emerge than simply through density-dependent interactions, for sure.
Thank you for the lectures. Really
Thank you.
Interesting, inspiring. I did not get the full extent of this enlightening, parental now is what's the other type of? I do not got the full extent of this event. This can you explain symbiotic relationships in the symbiotic cells? Can you explain the roots of Massey, I think, but can be applied to communities and other stuffs, more complex?
For sure, you can apply it to things that are much more complex than just this red and blue. You could apply this to any community you want. The only requirement, if you take this experimental setup, the only requirement is that you have some means of assaying a property of the community. But there are many possibilities. I mean, let me give you one example. Let's say you wanted to select for communities of microbes, let's say to add to yogurt, that would suppress Campylobacter, a very common foodborne pathogen. So what you could do is take a green fluorescently labeled Campylobacter and take a community of microbes from wherever you like, soil, gut. Introduce your fluorescently labeled Campylobacter into every single droplet, and then measure after a period of growth the fluorescent signal that comes from Campylobacter. And maybe you start with a large signal. And of course, you would reward the decision of which droplets are eliminated and which go on would be based on the extent of suppression of the fluorescent protein. So it'd be exactly the same, but different criteria. And that, if this is correct, would lead to the evolution of interactions and to a community of microbes that may be a consortium of many kinds that will build interactions that would be stable, that you could eventually take out of a drop and maybe even put into a flask and grow up separately, and it would retain those properties. Or, we also have technologies for spitting the droplets out and immediately encapsulating it in a polysaccharide polymer, and then that can go into your, into your yogurt. So you can do almost anything. And it is just about, again, imposing the logic of Darwinism. These communities become unwitting players in the selective process. And it will lead to the evolution of interactions. It will build symbiotic communities. The extent to which they become integrated, well, that needs to be determined. But I think there's every reason to think that this process that I outlined would build symbioses. Whether it even gets to the point of, engulfment of one organism by another, I mean, that's maybe seems fanciful, but as actually Daniel and I were talking about at coffee, what we are doing in the lab, of course, is imposing there's extreme imposition of these Darwinian properties. So selection at that second level is extremely strong. In nature, it is, and so we really might see the sorts of things that have happened extremely rarely in nature, because in nature, almost certainly, these extreme manifestations of Darwinian scaffolding never manifest. But that doesn't mean to say that's, they don't have to be extreme in nature. They could be minimal. You can relax. You could allow a little bit of migration between the droplets. So that blurs heredity. It won't stop selection working to some extent at the level of collectives. So I think using this kind of lab setup, you really could perhaps see some striking events. I mean, if you really wanted to evolve, take I think a good chance of getting an endosymbiotic event would be to take a non-photosynthetic alga and co-culture it with a photosynthetic bacterium and reward the collective on the basis of algal biomass, for example. Implement the same process. I wouldn't be surprised if an endosymbiotic event occurred. Because the thing is, it may be exceptionally rare, but this is the beauty of selection, right? If there is a process that will amplify that needle in the haystack event, then you will see it.
I'm a little bit confused about the actual definition of scaffolding. I understand there's this the notion of discreteness and selection acting on two different timescales, but I'm not really sure if I understand the whole concept.
So it is about making entities discrete. It's then about making entities or collectives of entities. So first of all, it's about so here are some entities. Let's put some more down here. So first of all, we put a boundary around them, like we did with the droplet. That could be a test tube. It could be something in nature. So we have discreteness. And more to the point, variation. This is different to this, by virtue of the composition of particles. The next part so scaffolding is all about imposition of these Darwinian properties, variation, reproduction, and heredity. We get variation, and it's discrete by virtue of putting boundaries around. We need reproduction, and we need heredity. And when they manifest, selection operates. So selection at this stage is not doing anything. But if what we can now do is allow a reproductive event, which we can bring about, so we can, we can split this in part, we allow this to grow up, we can split this, and so this has some sort of composition. And we could do the same with this one. Well, this has given us the reproduction. And then provided there is some resemblance between this state and this state, then we have a parent-offspring relationship. We have heredity to some extent. Now, the moment that we have variation, reproduction, and heredity, we have the conditions, the properties manifest that allow selection to work. So the scaffolding is simply the idea that we can provide a population structure by our manipulation of ecology that makes these collectives manifest these Darwinian properties. I'm not sure if I've really done that as clearly as I could, but so selection, once these three properties are met, any set of entities that have those properties will participate in the process of evolution by natural selection. So scaffolding is about externally imposing these. In the absence of us putting a boundary around there, those are just individual cells. We put a boundary around it and I said, I'm now going to make this collective and this collective participate in the process of evolution by natural selection. I have to offer two more things have to happen than just making the discreteness. They have to be able to reproduce, and the offspring have to resemble the parental type to some extent. That's what we can do. And then selection works. Selection, of course, manifests in the ways I've been talking about as a death-birth process. So again, what we've seen here, there are two entities here. Let's call this one A and this one B. B has died, it's gone extinct, but A has left two offspring. And we could draw that. We start off with two independent lineages. This is lineage A, this is lineage B, and lineage B has now gone on. There's an evolutionary process happening. A's gone extinct, B has expanded, it occupies two places. That process can go on. Variation arises within B's, and we can continue to produce this evolutionary radiation. So evolution now happens once we've got that process. So scaffolding is the idea, recognizes that these properties are not properties of these cells, and there are no meaningful groups here except those that I make by artificially putting a boundary around them and then forcing this reproduction. Yeah.
So, what I find is really interesting is how interactions, how you can pass on interactions in generations. So, is there a mechanistic basis, a deterministic basis on how interactions are formed? Because, if you could do that, if you think about even in communities that don't have a Darwinian stuff, to think about that interactions are maintained by a pressure of selection is very cool.
Yeah, can I just complement the question?
Yes.
Yeah, because you're imposing selection on numbers. It's a ratio that you keep constant.
And
that
what
reduces the development of some interactions.
Yes.
But it could be a competitive interaction, so that when you stop, one eats the other and it's gone. But it's always a cooperative interaction that develops. It's kind of mysterious. Why?
I guess that's because of the artificial selection at the level of collectives. Because if they don't find a way of converging on a one-to-one, they're eliminated. So if you didn't have that scaffold, you wouldn't see this sort of convergence to a kind of a, well, to say it's a mutual, it's the arriving at a constant ratio through some mechanism. And those mechanisms could be numerous. In this case, again, we've chosen for the theory part a very minimal set of criteria. But actually, that's what I was surprised when we first talked about this project, and Guy and I sat down. What was bothering me is the thought that we would have to have some parameter for stickiness, for example, which we would then have to put into the model. And then we've parameterized our model with the very thing that we want to see evolve, which, So what I really liked was this move to density dependence, which is a very simple kind of, the basis of very simple, it's a simple basis of complex interactions, but we could then start from a situation where there was no interaction, allow that to evolve, and see whether interactions do indeed evolve in our favor. in the system. So we've avoided an awful lot of complexification that would give many, many more opportunities, I think, for interactions of a wide variety of types. I mean, Joshua was pointing out you could have interactions from transfer of DNA from one to the other. That would be, that would be a winning way to solve the alignment of reproductive fates.
To do this experiment, it lasts at least more than 1 year to find these 9,000 generations.
Yeah. Any students? There are positions, actually, if you want to work on this topic, both theoretical and experimental.
And something that you found is that the growth rate changed from A to B, B to A.
Yeah.
And this was perhaps the main parameter to find the equilibrium that you forced with the bottleneck.
Yeah. Well, and the change in growth rates, we hadn't appreciated this to begin with, but it makes sense, was necessary to allow the establishment of density-dependent interactions, which is kind of nice. So although there was the potential for density-dependent interactions right from the outset, we didn't see the emergence of those until growth rates had increased to the point where these cells now spent a significant amount of time in stationary phase where those density-dependent interactions can emerge.
And this is the significant property that you could measure to find this.
Yeah.
And the last question, one of the last perhaps plot that you put is the ODE versus the experiment.
Yeah.
And there is a little difference before they found the steady state.
Yeah.
So your ODE is wrong. It's not a lot of Voltaire because there is a lag between the experiment.
I will have to direct that to Guillaume. Yeah.
Have you studied this, or will you do in the next months?
I have. Yes, there is an extensive supplement where he has looked at the fit between his ODEs and the simulation results, and actually I have that here. You'll be welcome to have a look, and it may answer your questions. Yeah. But actually, with regard to doing the experiment, I mentioned to someone at the break, what we've gone away from is the machine that I showed you the video of, to a very cunning design that where we have 2 coils. There is a channel, they're connected, tiny channel, and this has growth, and this is a separate coil which is uninoculated. And using lasers to determine, on the basis of the refraction of light at the tips of the droplets, we can precisely know where the droplets are. By controlling pressure, we can stop the train. And so what is possible is to say, this is a winning droplet, and it's been chosen to leave offspring. So this is now brought to this position here. This one here is brought to this position here. And then we squeeze the tube, and a sample of bacteria flows and inoculates this. Then we allow this to move along one. So this one now becomes here. We repeat this squeeze and then move the whole thing along. This can be done. We can do 1,000 droplets in about 10 minutes. So, and what it also means is that we can have this event taking place every 12 hours if we want. And people don't need to stay up all night. So it makes actually doing the experiment much more realistic. The problem with the sorter that I showed you before, and this it sips and then it sorts, the sorting process is too slow, and it's also the engineering behind it is complicated because you have a constant flow of droplets, and if you ever have to move anything other than the exact same time interval, then you've got the problem of coordinating a longer time with the fact that the droplet, and it goes crazy. Yeah. So we can do the experiment.
How many bacteria can fulfill in a droplet, the maximum, without stress?
I mean, we can control the time that we squeeze, and we can control the size of this. And these droplets are now up to.
In your first experiment,
yeah.
that you show, you'll have a big droplet, and the bacteria can grow.
Yes.
How's the maximum number that you can?
About 1 million per microliter.
And the droplet volume is?
In that machine I showed you, we're using about half a microliter. But here, with this revised, we've got a bigger train, and the droplets are up to 10 microliters. So there we're up to 10 to...
You grow without stress.
I think so. Yeah, they're well oxygenated, they're well mixed, because they continually... Yeah. They grow better in here, much better than here than in a microtiter plate, for example.
I understand that this selection was made possible by an ensemble-like setup. So you had a lot of copies, and these copies could, when they reproduced, you could occupy other copies. But in a time-like framework, where, for instance, you had just one big environment in which communities could turn over. Would this kind of a reason be possible and be applicable in that sense?
So in that large community, there's no structure. Is that right? You just sort of got a well-mixed environment.
Yeah, in my mind.
So
yes.
it won't work. Yeah. So this is I think an important realization too. A lot of origins of life work at the moment. Some extremely interesting chemistry. But for the most part, it's being done in the equivalent of a bucket, where there is no spatial structure. And there is basically no opportunity for any amplification, so no opportunity for selection. So there may be a brilliant chemical reaction that occurs that really could set up some autocatalytic process. But if it's immediately after first realization diluted or it's not compartmentalized, it will never experience the amplifying effects of selection. So here, that discreteness is critical, and it is full stop for any Darwinian process. If you've got constant mixing, even if we did this experiment in our droplets, but every generation we pulled everything into a big test tube and then re-established the train, we wouldn't see any selective response. There would be no parent-offspring relationship, or absolutely minimal. Yeah. So yes, good question in the sense that it makes... Yeah, you need that spatial structure. You need that discreteness.
Okay, so I think we should stop.
Okay.
So we come back here in 15 minutes for the next lecture. Okay, so let's thank Paul again.
Thank you. Thank you. Okay. Yeah.
Can I ask you another thing?
Yeah.
You also